Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia. Please allow me to welcome you to this edition of our 2020-2021 Foundations of Biomedical Data Science Seminar Series. This lecture is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and by a grant from the National Institutes of General Medical Sciences. It is my delight today to welcome our friend and colleague, Dr. Neda Jahanshad from the University of Southern California in Los Angeles, California. Neda Jahanshad is an associate professor of neurology at the Imaging Genetics Center, which is part of the Stevens Neuroimaging and Informatics Institute at the University of Southern California. She holds undergraduate degrees from Johns Hopkins University and her PhD from the University of California, Los Angeles. Dr. Jahanshad has over 10 years of experience in analyzing brain scans from large groups of individuals and comparing magnetic resonance imaging scans of people with and without brain disorders of all ages. Much of her research explores the brain as a network of interconnected pathways using diffusion-weighted MRI, a technology which reveals physical connections between different functional areas of the brain. Dr. Jahanshad also directs the Laboratory of Brain E-Sciences at USC. LOBES, as it is called, is a collaborative multidisciplinary team which combines multimodal brain imaging techniques with advanced computational modeling approaches and information sciences to better understand the brain and reliably identify risk factors which contribute to its decline in aging. The lab aims to ensure that their tools and approaches and findings bridge international boundaries for broadest possible applications. Dr. Jahanshad is a key developer for the Enigma consortium, which brings together hundreds of researchers from around the planet. These researchers work together to identify the genetic environmental risk factors which make the brain susceptible to disease. Dr. Jahanshad's presentation today is entitled Advancing Neuroimaging Genomics with Continuous Team Science. And as always, we are streaming this lecture live and for recording via YouTube. And if you are watching via YouTube today, thank you so much for joining us. Also, our specially selected 2020-2021 Biomedical Data Science Innovation Lab participants are strongly encouraged to submit any questions for Dr. Jahanshad via the chat feature in their Zoom sessions. I will synthesize these questions and I'll ask them on your behalf during the last 10 minutes or so of the lecture. And so, Neda, thank you so much for doing us the favor of presenting your work today, and we are excited to hear your presentation.
Thank you so much, Dr. Van Horn. I'm excited to be here. I'm just going to turn off my video so it's easier for me to give the presentation, and I'll turn it back on for questions. Well, thanks everyone. As Dr. Van Horn mentioned, my talk today is called Advancing Neuroimaging Genomics with Continuous Team Science. I have some NIH funding and funding from Biogen that's unrelated to the contents of this talk. So as I'm sure you're all well aware now, especially towards the end of this seminar series, there has been a replication crisis in brain imaging. Hopefully that, I should silence my phone next time. So what does that, where does that come from? So just a little bit of background. Most of the neuroimaging studies that sort of culminated in this replication crisis, a lot of them had been in very small sample sizes, or now we're seeing as relatively small, between 20 and maybe 200. And they were only conducted within single studies without independent replications. And some of them didn't even address multiple testing issues well. So when going back and trying to rerun a lot of these tests or trying to replicate these results, claims in many papers did not hold. In addition to some of these technical issues, there's a publication bias that often prevents null or contradictory findings from being reported. So there's a potential inflation of effect sizes in these literature-based meta-analyses where you're really just pulling together findings that were significant or successful. Now, it's obviously one of the reasons behind these small sample sizes is because, at least in brain imaging, scanning is pretty expensive. It's approximately $600 an hour just for the scan alone. And you need oftentimes about an hour per subject. So there have been several initiatives over the last almost 2 decades now, where there are multi-center initiatives that aim to coordinate data collection across many sites and ensure well-powered studies. And this involves some prospective data harmonization efforts, let's say imaging phantoms, and ensuring that the protocols across the sites are the same. These are all exceptional resources for scientific discovery. But on the other hand, what happens to all the data that was not from these multi-million-dollar multi-center initiatives, all the studies that were collected before, all those smaller individual studies? So at the time, a lot of people were actually being funded to study some of the same questions in regards to brain development, psychiatric disorders, aging, dementia, oftentimes in different population. And everyone would have their own protocols and imaging protocols, or maybe different functional tasks. So the one idea is can all these individual smaller studies that were funded to do the same ultimate analysis, can they be merged for more powerful, reliable findings? And could they be merged with each other or with these newer multi-site initiatives, and how? So that's sort of the basis behind the Enigma consortium, which I'm a part of. So Enigma stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. And I'll get through why this acronym is not exactly what we do anymore. So Enigma started with the idea of genetics, and especially because neuroimaging genetics in particular were very susceptible to false positives. And you could say, well, why is that the case? We've had these neuroimaging genetic studies for two decades now. So we know some of these candidate genes. So I've just here taken a snapshot of some papers, and this is not to call anyone out if they've been involved in these papers. Some of them I've been involved in. It's just at the time we thought we knew a risk factor for brain decline. So, but we actually hadn't double checked. A lot of them had weak or no statistical correlations. There was a lack of replication, lack of power. And if you do a power analysis, you need 800 people to detect a 1% effect. And this is a very big effect for a SNP or a single nucleotide variant or genetic marker with 80% power at p less than 0.05, sorry. So if SNPs explain just 1% or less than 1%, then why are these imaging genetic studies being done in less than 100 people? And that was because we were doing underpowered candidate neuroimaging genetic studies. So we will see that a lot of these were in about 100 or less participants. And sometimes they would even test multiple factors, but not correct for them. So if you're testing 5 domains, you should correct for 5 tests. So we'll see why there was this sort of push towards, or this decline towards the crisis, because we weren't looking at things properly. So Enigma began in 2009, initially to identify genetic loci that reliably contribute to brain structure. And the reason we focused on this genetic aspect is because genetics offer this direct link to the underlying biological mechanism, which means that they are easy targets for treatment. And at the same time, you have the imaging marker for monitoring and assessing the efficacy of any treatments. So it's important to keep in mind that when you're scanning the genome, you actually have about 1 million independent common variants. So you're not just doing one test, you're doing 1 million tests. So you need to correct for those 1 million tests and set your significance threshold appropriately. And that would be 5 times 10 to the negative 8 for a single genome-wide association study. It's important to keep in mind that your power doesn't depend on the test that you're doing. So yeah, MRI is more expensive to collect than, let's say, classifying someone as having a disease or not, but it's not more powerful. So you still need the sample size. And to illustrate that, this paper looks at the number of subjects needed for a given number of tests with respect to the percent variance explained. And as I mentioned, a single nucleotide genetic marker often has less than 1% variance explained. Oops, this got moved. So what we would have is actually around here. If we're doing 10 times 10 to the 6th test, the brain should actually be around here. So we have about at least 10,000 individuals that we would need for a successful genome-wide association test to have the power for it. But how are we going to get 10,000 scans? We, as I mentioned, we're going to try and pool all this data from all the smaller studies from around the world. And as I'm sure you're aware, a person scanned in one scanner is not going to have the same exact MRI image as that same person scanned in another scanner. So this is an example of one person scanned at a 1.5 T scanner, and then again at a 3 Tesla scanner. So you can see, if you're familiar with looking at brains, you could probably guess that this is the same person, but you can clearly see that the brain structure has different contrasts. So like the gray and white matter contrasts are different. Some structures are easier to see than others. So we have these issues that we're trying to work through. And our first approach is this analytical harmonization. So we might want to actually embrace this variability in the acquisition. You're not going to necessarily want to have your finding depend on a given scanner. If you're trying to develop a medication or trying to monitor brain tissue atrophy, you don't want that quantity to have to depend on a single scanner. Otherwise, everyone in the world who might have a disorder related to your treatment or the biomarker would have to go to a particular location, and we know how difficult that could be. So what we decided to do is just to try and embrace that variability and harmonize the analysis plan instead. So kind of select certain metrics to analyze, the same across everyone, ensure that quality assurance is done similarly across sites, and have the same statistical workflow. And this works exceptionally well for this coordinated meta-analysis that we did. And to pool all this data, we want to ensure that the metrics that we're extracting, the brain metrics, are derived reliably from scans regardless of how they were acquired. Again, we're trying to embrace this variability, but we do want to make sure that whatever measures we're extracting are reliable, regardless of of the scanner. So before we distribute our protocols and let people know what measures we're trying to extract, we need to test those measures. Are those measures extracted reliably? And are they reliably reliable? And by that I mean, again, does it depend on a single scanner, or can we reliably extract this measure across different scanners? And we'll have often 4 or 5 sites beta test any protocols, make sure that everything works. If there are any frequently asked questions, we'll make note of that and try and troubleshoot it and improve our protocols. And once the quality assurance measures are in place, then we can send them out. So one tool to start with, many brain imaging sites around the world are familiar with these common tools such as FreeSurfer for cortical analyses. So before we say, hi everyone, run FreeSurfer, we make sure to test whether the measures we're looking at are reliable. So is there high interclass correlation? So if you do a retest analysis at different scanning sites, is it reliable regardless? And do we have quality control protocols? So we put that into place. So ways that individuals can look for errors in the outcome, like make sure the whole cortex is extracted. So we have created cortical quality control protocols that we've also distributed alongside the analysis plan. So we have several imaging protocols. The first one I mentioned is based on FreeSurfer, but there are other ones based on diffusion tensor imaging or more detailed analyses of shape. We recently, a group in Germany that we work closely with has developed a harmonized analysis of fMRI processing, which could be used for resting state and also for task-based fMRI. So going back into the FreeSurfer protocols, I'm going to go through a recent example of how we use this and to pool all the data. So the latest genome-wide association study we did was entitled The Genetic Architecture of the Human Cortex, and it was published in Science last year. So we asked the question, can we identify these genetic variants that are associated with just normal variation in cortical surface area thickness as a whole, and also these regional patterns? So at each site, we ran the GWAS of cortical structure. So again, we're making use of the not only the data sets at each site, but also the computational infrastructure. So we're doing this distributed analysis where each site does the statistical analysis, does all the computation, and then the results are all pulled together. So once all the sites have done the analysis, they send only their results, not the images or the genetics themselves, to a centralized location where we do the meta-analysis, and we do some quality control tests on top of that. And then once we pull all the data together and make sure that there are no quality issues, we found over 200 genetic regions that are associated with either one trait, surface area, or thickness, or those traits in particular regions. And we found that actually most loci were unique to a particular brain or trait. So we do see this regional patterning of effects. We saw that the cortical, the genetic architecture of the cortical measures are actually pretty different. So surface area was a little bit different than thickness, with surface area finding several more regions of genetic association. And within surface area, actually, we see this regional patterning where we found a lot more genetic associations with areas of the occipital lobe than some of the other regions. So we have this polygenicity of the occipital lobe, where there are more genes associated with that, or genetic loci. And you can actually take the full genome-wide results, so not looking at the significant loci only, but really the across all 1 million independent regions, and correlate those with genetic results of other traits. And we actually see a positive correlation between, as you might expect, greater surface area and greater educational attainment, and this is a genetic correlation. And also a negative correlation between traits such as ADHD and insomnia, where lower surface area correlates, or genetic risk for lower surface area correlates with genetic risk for ADHD and insomnia. And interestingly, there's a positive correlation with Parkinson's disease, where genetic risk for higher surface area also correlates with genetic risk for increased risk of Parkinson's. So this reproducibility is actually an issue across these biomedical fields, not just genetics. And our goal is not to just focus on genetics, but to ensure that this reproducibility is something that we can evaluate across all of the neuroimaging sciences. So we extend these methods far beyond genetics to look at a series of disorders, diseases, and various conditions, even in normal development and healthy individuals. So we are using these protocols that are similar across these working groups. So the same quality control protocols, the same analyses. We are able to take the results and compare them to each other. So you get this interesting overview of how different disorders, for example, might have different effects on the brain. And these are all conducted using similar protocols. And each one of these analyses had over 1,000 participants. And as I mentioned, it's not, our work is not just limited to free surfer and cortical surface area or thickness, but we've also done some work with diffusion tensor imaging. And we're not only looking for reliable effects across the data that we're using, but this was an interesting application where, after we published this paper with I think over 30 sites participating in finding globally reduced or lower FA in schizophrenia participants versus controls, a separate consortium, a separate multi-site consortium with only Japanese sites found very similar results. And we were able to look at these side by side, and the other consortium also used exactly the same imaging protocols which we had made public. So that's kind of a brief overview of a lot of the Enigma work, but there's plenty more. So if you're interested in any of the conditions or disorders, or studying any of the traits that you saw in that plot, do reach out, because there's always room for additional analyses. And that sort of gets into my next aspect of how we're extending some of these work, and that's to look at really the continuous portion of this. As I mentioned, every time we're presenting work on Enigma, there might be someone who has a data set that they would like to contribute to these ongoing analyses. So we have this continuous analysis, where as more and more groups are joining, our sample size is growing, and with greater sample size, you have more power to detect effects. Sometimes you can detect sort of the same effect, but with more power. So this is an example of our first study looking at doing a genome-wide association for hippocampal volume. We had about 8,000 individual scans across all the data sets. And this was our top hit, was just below the significance threshold. In a second analyses, there were 5,000 more scans that had been contributed from many sites around the world. And that top hit was now past that significance threshold and was significant. And very recently, the data, the sample size doubled, and you can see this evolution of significance in these findings. As we're continuing these analyses, a lot of them are repeated. You're seeing this sort of evolution in results, and not all of them are in this direction. Sometimes you'll see a result that might have gone away once more samples were added. So it's important to have this evolution of knowledge and refining our understanding of these tests that we're doing. So we're developing this sort of infrastructure to accommodate for these continuous analyses. As there's more and more data being contributed or even collected, can we do what we're doing in a continuous manner, sort of automatically, actually? So can we, so we're working with researchers at the Information Sciences Institute at USC to develop this sort of workflow. And just to give you a little bit of background, so this is a new infrastructure that we're calling NeuroDisc, and there's a website that will be made public very soon. We ask a hypothesis, which is a question that we want to test, and this is linked with a line of inquiry that will look for data in this wiki that we've developed, and the wiki has certain properties. It has a given ontology, and the data sets are annotated to include features for knowing whether or not that data set would be included in a particular hypothesis. And so we can look for data that would match our hypothesis, and then we have workflows, let's see, analyses, statistical analyses in a separate system that gets called, and all that together allows us to continuously update our knowledge. So here's an example. Let's ask a hypothesis. This is based on the results of the Cortical genetics paper that I mentioned. So let's ask, for example, is the effect size of this genetic variant, which was actually our top genetic variant, on the surface area of the precentral cortex, is it associated with age? So that triggers this line of inquiry, which says, okay, we're looking for the effect size of a particular genotype on a certain brain imaging trait, and we're looking to see if that's associated with a demographic, which in our case would be age. So that triggers a lookup of our wiki, so our meta wiki, where we have all the annotations, and also the statistical analyses gets run with all the data that it finds. And so this allows this sort of continuous evolution. So if you miss your chance to participate in a study that was published, we have the protocols in place in terms of the imaging protocol, so you know to run those. You have the statistical protocols also, which are made public, and those results can get integrated even if the data doesn't get shared through And by the data, I mean the individual subject level data, which is often sometimes shareable, sometimes not. So we work under the assumption that it doesn't always need to be shared at the individual subject level. So even if that is not the case, you could still contribute results and be a part of this continued workflow. And as an example, we ran the results of our again, cortical genetic paper with and without the largest sample that we had, which was the UK Biobank. So if that data set got added in later, we have an evolution of the results. So these are the forest plots, which show each individual cohort that participated. And then after we add the UK Biobank, you see this additional row here. And in our example of looking at the association with age, or mean age of the cohort, we plot mean age on the x-axis and the effect size on the y-axis, and you see the trend line sort of evolve after the addition of new data sets. So we also have other extensions and initiatives, including statistical and image harmonization for beyond meta-analysis. So if you can share the individual subject level data, what can we do? So beyond meta-analysis, that's exactly what I mean. Basically, you're no longer meta-analyzing statistical results from individual groups, but you have individual subject level data from all sites, and you have essentially a mega-analysis where all the data can be joined, and you can look for, it actually allows to look for more than, for let's say rarer occurrences. So in this case, in this epilepsy diffusion paper that we published in Brain last year, the individual subject level diffusion measures were all pooled to look for effects of subtypes of epilepsy. And some of these subtypes are not as common as others. So you might get, let's say, 3 or 4 individuals within a particular cohort that have this subtype. But when you're in that 3 or 4 is certainly not enough to do a well-powered statistical test, even if it will be pooled with other data later, or other effect sizes later. So by pooling these rarer subtypes together in one big cohort, then you can do a more powerful test. You have to, in some sense, harmonize the data sets to account for the scanner variability that we talked about earlier. So in this case, we did a statistical harmonization where, if you can see here, before the harmonization, the distribution for each site is a different color in this plot. And you see how they're basically all over the place for FA. Every site has its own distribution. And for those familiar with diffusion imaging, you know that that makes perfect sense, because these values are essentially only dependent on the acquisition of the image. So after this combat statistical correction, we see that all the data sites, all the sites have their data distributions with similar means and variance. So that's how we were able to do these analyses and adjust for these site effects. So as I explained, the meta-analysis is where the stats are run at the individual sites and the results are merged. And it does allow for the subject level data to remain local. So if it cannot be shared, then that's fine. And then on the other hand, the mega-analysis, the subject level data are pulled together, and then the analysis is run on the cohort or the huge data sample as a whole. So we are, harmonization is actually a very active area of study. This is another example of how statistical harmonization can be used. We have a paper out on medRxiv looking at different stages of Parkinson's disease. And again, not all cohorts collect, or are looking at Parkinson's patients or people with Parkinson's at all these different stages. So you might have lower frequency of certain stages at certain sites. And by pooling all the data and doing harmonization, trying to account for these site effects, we're able to focus our statistical analyses on these subgroups of individuals at different honing stages. But harmonization is actually a very active area of study. And there are some deep learning methods that are now being used to try and remove the site effect. So one area that we've been working on, Mengting Liu in my group, has this new method based on star GANs 2, where each, if you see here, the static images represent the original image at the top row. And then the bottom row represents that same subject image harmonized to another scan. So what you're seeing kind of move up here, this is what we're harmonizing to. We're trying to match the style of any other image. So you can see that the contrast changes, but we're not adjusting structure at all. So it should still be the same individual, but perhaps look like it was scanned at a different scanner. Another approach could be, and this is again another active area of study, whether we can derive acquisition invariant imaging traits. So if you were to extract the mid-sagittal corpus callosum from individuals scanned at different scanners, the thickness might be very dependent on the voxel size of the image, but the curvature might be something that you could extract reliably across scans. So this is something we're actively working on in my group. Alyssa Zhu has published a SIPIM paper a couple of years ago, and now we have a deep learning approach for this extraction and deriving these measures of curvature. We're also working on better methods for extracting information from diffusion imaging as well. And Talia Neer in my group showed that fractional anisotropy, or FA, derived from the tensor distribution function, which essentially is looking at all the possible tensors that you can fit to a given voxel instead of the best one, which is usually done with the standard DTI. That actually gives a more homogeneous distribution across sites than the standard FA alone. So that was interesting as well. And all this harmonization and all of this ensuring common protocols and statistical approaches and quality control measures across sites also allows us to ask questions across across these groups that maybe we couldn't ask before. So one nice thing about this is that it does open the door for looking at cross-disorder or transdiagnostic effects. So if there are symptoms or outcomes that are similar across different conditions or disorders, maybe we could look across those and not have a disorder-specific analysis that we're doing. So suicidal thoughts and behaviors are common across many mental illnesses, and we are using these statistical and image harmonization efforts to address this and pool the data together and account for the fact that there are there's a low base rate of suicide attempts in a given collected cohort. So even though it's rare, it's obviously a very big problem. So we need these harmonization methods to be able to pool the data, to be able to ask these questions and have the power to answer the questions reliably. So this is one initiative that we're currently working on, is the Enigma Suicidal Thoughts and Behaviors group, which again is not limited to a particular disease or disorder, but is trying to look across all of them to be able to identify the neurobiological signatures of suicidal traits and other factors that might associate with those. So right now, we have been able to look across various disorders, including schizophrenia, bipolar, major depression. We do have some OCD and PTSD participants as well. And we're not just comparing individuals who've had suicidal attempts with healthy controls, but now we have clinical controls, where these are individuals that have the same diagnoses, but might not have made a suicide attempt or do not have suicidal ideations. So this is an active area of work, and I won't present the results because these are being updated regularly. So in summary, sort of this international multi-site collaboration that we're doing has really given us an opportunity to work in a well-powered environment for neuroscience with computational efficiency, especially if for the meta-analysis studies where you have the computation being done in a distributed manner, and also is an opportunity to really look at this open science and sharing of information, and to collaborate to ensure reproducibility and get these well-powered results. And of course, that's related to the team science as well. In recent years, it's really been opened up new, I think, technical questions as well. How do we best harmonize the data, and how do we advance neuroscientific discovery? There are hundreds of people in Enigma, and I would want to thank them all, but I don't think you want to sit through that. So I will refer you to this recent review paper if you're interested in learning more. This is published in Translational Psychiatry, where it describes the last 10 years of our work in Enigma. And thank you to the lab members and to the folks whose results I did present today. And thanks. And with that, I'm happy to answer any questions.
Netta, thank you so much for summarizing Enigma for us, and all the work that you guys have been doing through Enigma. It's a fine example of team science writ large around the planet. It's really a great kind of group resource is just a great example of that kind of done at scale, if you will. I like the idea of, you kind of touching on the notion of publication bias. This is something I've looked at myself, where, we've had this as a problem in neuroimaging since its inception, where, I can remember the early days of neuroimaging and the certain software packages who will remain nameless, where they hard coded the number of subjects that you are permitted to use in their software at n equals 6. And when you were trying to do this, the groups that would use the software or software like that with these small sample sizes, they were just very prolific. So they're flooding the literature with these small sample sizes. And of course, they're only reporting on the stuff that was statistically significant. So early on, we came out of the chute with a publication bias problem. And it's interesting to see Enigma taking this really kind of global planetary, if you will, approach to bringing people together around this. I have a couple of questions. One of them is, have you noticed any site-based differences? I know that the whole effort is to kind of grab all the data and pool it into one big sample, but I'm also kind of curious about, do you see, say, country by country based differences, site by site based differences? Are those evident, and how do you adjust for them?
There are huge differences. And one example, I think that it sticks out in my mind is this recent work that we published early this year on our HIV collaboration. Mm-hmm. So we have many sites in the US. We had one site in Europe, and most of the US-based sites were majority male. Between, I'd say, 70 to 100% of the participants at each of the individual US sites were male. Now, we also had a cohort in South Africa, and that one was largely female. That was the only one that was largely female. So we see these differences that are not only based on the imaging acquisition at these different sites, but the recruitment. And I think that is an important thing to be aware of in these harmonization approaches, because a lot of the harmonization approaches attempt to statistically match the distributions, but maybe they're not meant to be statistically matched. Maybe there are biological differences in these cohorts that should be taken into account. So this is a very good question, because it's an open area of research, and it's a challenge that we're facing. Is a lot of times we might ask what proportion of the cohort is male or female, but there might be information that we don't ask and we don't know about. But there are clear differences between like ethnic groups or countries or just it's. How do we list the things we don't know?
I think that, it's such an interesting thing, because with the way in which Enigma has approached this, which is, laudable, because it's clearly been very productive, in maybe a more traditional literature-based meta-analysis, where you're gathering, trying to estimate effect sizes from particular studies in a literature, you can also incorporate the study kind of factors that went into the creation of that effect size, right? The end result. And it could be diagnostic criteria differences. For MRI, it could be TRTE differences or contrast related differences. It could be things like the proportion of males and females in the sample, as you point out. In meta-analyses I've done, these are somewhat dated now, but we found a statistical correlation between the year and the size of the effect and the year in which the study was done. And apparently in the example I did ages ago, apparently this is a common thing, that the early effect sizes tend to be larger. And as you get better at doing those studies and you incorporate more controls to them, that the effect sizes get smaller. One additional thing, which was kind of ironic, I suppose, from an Enigma point of view, is that we found that the size of the effect was also inversely correlated with the number of authors on the paper. Which we thought was kind of interesting from a sociology of science point of view. But anyway, it would be very interesting to explore some of those study factors, because you may have, like, country by country differences in, for example, how Parkinson's is defined or how Alzheimer's is diagnosed or what have you?
Absolutely. I agree. There are certainly differences, but at the same time, one of the things we're trying to do is embrace those differences. We're not trying to solve
absolutely.
Alzheimer's in one location only.
Absolutely. I mean, hopefully these things are, they're random enough that you can average over them and be able to do okay. One of the things which I just, correct me if I'm wrong, but the genome-wide association work that you're doing is that's largely based upon SNPs, SNP-related genomics. Is there any opportunity for Enigma to adapt to more full genome sequence technologies as they become more readily available?
So there are efforts looking at rare variants, for example, and that or copy number variants, or like 22q deletion is its own working group. So we do have efforts outside of just the full genome-wide screen. Whole genome to really make the most of that entire genome screening, you would and look across it in an unbiased way. We do not have the sample sizes in neuroimaging to do that yet.
Yeah.
There might be ways, as I mentioned, to kind of look at, maybe use the whole genome to look at regions around common variants that we found significant, to look for causal loci or really sort of pinpoint some of the genetic factors. But in terms of screening whole genome, I'm not sure if there are fields that are there yet, even
not just yet.
Neuroimaging.
That's the next grant. What you mentioned, the word commonalities, which is kind of interesting. I'm curious if you guys have a lot of the analyses and all the working groups. They seem to be very disease specific, and I think you sort of hinted at this in part of your presentation about trying to do it irrespective of disease and look for, say, commonalities across these different diagnostic distinctions to see if you can't almost like anneal different disease kind of profiles into more common ones. Does that make sense? You know what I mean? Where, for example, two diseases may be somewhat similar, so similar enough that their distributions overlap in some way. But I was just curious if you guys have attempted to do anything like that. Yeah,
there are some a lot more cross-diagnostic work now,
Right.
and sort of transdiagnostic, as I mentioned, with suicidal ideation and attempt work. But there are efforts to try and look at symptom profiles across different disorders. Like there's currently an effort to pool the data from groups that have studied depression or major depression, bipolar, and schizophrenia into one, and look for effects of, let's say, antidepressants across these individuals, or look for common profiles, look for what distinguishes bipolar from major depression in terms of brain circuitry, and there are current efforts that have really over the last year or two really taken off once the sample size were large enough to work with. So.
What clearly Enigma is doing a great job at identifying these effects, and linking things to particular gene profiles, and obviously the sample size just keeps getting larger. Is Enigma the results from Enigma, these results of these studies? Do you know if any of the studies are being used to inform clinical treatments for any of these disorders?
That's a really good question. I think there are efforts to try and get there. A lot of our first sets of papers were really to establish that this data could even be pulled together, and that we can look for these common trends across, like you said, the different criteria for diagnoses, or the different definitions of certain symptoms across the world. And now that we're seeing that is possible and that is feasible, that's now there are efforts to answer, see if we can try and answer the next questions, is why would some people respond to certain treatments and not others? What does that treatment, do to the brain? And I think that will open the door for clinical trials and more translational clinical work.
You mentioned mega analyses, kind of pooling not just the derived metrics, but actually going and getting the actual scans and doing a big combined massive analysis. What sort of resources would you expect, or maybe you're experienced with, needing in order to carry out those kind of analyses? Is this like a cloud only based thing that you can only like send it to Amazon for? Can you do this on your laptop? What do you need?
So it depends on the type of mega analysis. So I think by mega, I don't necessarily mean the scan as a whole, but it could even be the derived features. The meta was more the statistics, but the mega could be like just having hippocampal volume across all the individuals. So I think a lot of people have been making proposals based on their resources, right? If they don't have the computational ability to have 10,000 full sets of diffusion imaging, then that's not what they're going to work with. But yeah, there. I think this network and the trust, and then also the push from the funding bodies in some countries and not others to share data has really helped sort of drive some of these mega analytical approaches.
Which is kind of brings me to my kind of last question. I'm very interested in your comments on this. Enigma is this fine example of team science in action writ large. And I'm curious what sort of lessons learned you might be able to share with us about how it's gone. Like what's gone right? What are the strengths? What are the weaknesses? Or maybe room for improvement in big scale team science like this?
So I think you have to go in, making a proposal, knowing that it's going to take a lot longer to do the work than maybe you think. There's the bottleneck is always going to be the slowest person to respond, and the person to send the data wrong or maybe not in the correct format that you asked. And I think what it has really helped us understand some common errors that we look for. So if we say, all right, everyone code a group of patients as one and the controls as 0, there might be some group that will do it the other way. So how can we check for that using stats or different profiles? Males and females were one which had been coded one way in one group and then another way in the rest of the groups, and we were able to look for mean trends in intracranial volume to understand that the coding had been mismatched. So I think knowing that you have to have another level of quality control when you're doing these pooled analyses I think has been a benefit, because it provides more assurance that what we're doing is meaningful and has hopefully less noise than it would otherwise. I think we've all learned over the past year that a lot of this, you don't have to do research with the people in an office next door to you, but you really are able to work with people all over the world, and it's just as easy as working with people at the same university. So I think we perfected Zoom way before most people had heard of it.
You're early adopters.
Yeah.
Well, Meta Jahanshad, thank you so very much for sharing your work with us and all of the interesting things you guys are doing in team science and meta-analysis and full genome-wide association studies in neuroimaging. Super exciting, very pioneering work. And thank you and all your team members for do the work that you guys are doing. I want to thank everybody who tuned in today, and we'll look forward to seeing you next week. Thanks, everybody. Have a good weekend.